Yeah, keep asking questions. All right, microbiology. Microbiology is the study of microorganisms, their characteristics, adaptations, physiology, and function. Okay, so it's very much like biology, but just for microorganisms. Here you will examine the role of diverse microorganisms in all aspects of life and the parts and functions that they have. You may look at different areas of microbiology, such as environmental microbiology, food microbiology, medical microbiology, microbes and biotechnology, virology. The list goes on. Okay, so there are different areas of microbiology that you will look at. What it looks like, usually you have around 2 to 4 weekly lectures where there'll be the delivery of the main content that you'll be assessed on. You'll also have tutorial classes where you'll go through practice questions and problems, and it'll be application based. So in your lecture, you'll learn about this content, learn this about food microbiology, for example, and then tutorial classes, you'll look at a real life issue and apply your knowledge and content to it. Labs will be application via experiments. So you'll be doing experiments with different microorganisms. The assessments are usually you have a weekly or fortnightly quiz, just like for anatomy, which will be likely multiple choice. You will have a midterm and an end of term examination and a final examination. So don't confuse the end of term with the final examination. Usually for me, because I have 10 weeks, I usually have a exam during week 5 or week 6, and then another one in week 10. And then I will have my exam from outside of the term in my exam period for the subject. You'll notice in anatomy and physiology, you don't do any like essays or any hand-in kind of things. It's all kind of quizzes. For microbiology, you will have something hand-in. So it might be a literature review where you have to look at a medical study and review how well it was done, its limitations, what it is actually talking about, what the results were. A project report where you have a particular topic, and you or a group have to research it and formulate an inquiry question and answer it. You can do a blog post, which is something I did last year. It's basically like a lit review, but it has pictures in it, and you can ask, like, it has like a captivating heading, but it's essentially a literature review. And there are other things, essays, but these are the 3 biggest ones I've seen: literature review, project reports, and blog posts. It's effectively a research paper on an aspect of microbiology. And then you might also have a peer review count, which will be a part of the total weighting. So let's say your lit review is worth 25%. 5% of that 25% is comes from peer review. So of that 5%, stay with me now. So we had 25% for the lit review, 5% for peer review. What is peer review? It's effectively where you analyze and mark and assess your peers' work and they mark yours. So of that 5%, 2.5% might be you marking others and how close to the teacher's mark you were able to get. So let's say you gave someone 20 out of 20, but the teacher gave them 10 out of 20, compared to if you gave them 15 out of 20 and the teacher gave them 14 out of 20, you're going to do better the closer you get to the teacher's mark. And then the other 2.5% will be people's mark of yours. So if they gave you 10 out of 20 or 11 out of 20, it will affect your mark as well. I don't really like having peer reviews because some people are really harsh, and even if they're not close to the tutor's mark or the lecturer's mark, they can still influence your results. But I didn't come up with that. It's what a lot of unis do. So just know that you can also have a peer review aspect. Okay, so let's ease into some of the microbiome content. Again, I'm doing this to show you the kind of stuff you'll be learning, help teach you some of the stuff ahead so you're extra prepared, and we'll be doing some practice questions. There are 3 main parts of microbiology: bacteria, eukarya or eukaryotes, and archaea. Okay, those are the 3 branches of what we call the phylogenetic tree of life. Okay, we are eukaryotes. Okay, we're complex cells, we're multicellular. We also have archaea and bacteria. Okay, we will mostly, or you will in microbiology, focus on these 2. Okay, some brief concepts. Anabolism versus catabolism. Catabolism is breaking down molecules to release energy. Anabolism is building up molecules, which uses energy. Okay, so catabolism, Siri again. I don't know if you guys can see that, but Siri keeps coming up. Yeah, so catabolism, we have a molecule, we break it down into smaller units and it releases energy, right? Because this big molecule had a lot of energy. Obviously, when we break it down, the energy is released. Anabolism is the opposite. We have a bunch of small units, we build it up to make a big one, and of course, if we're building something up, we require energy. How does this happen? Through metabolic pathways, okay? Metabolic pathways. There are a plethora of metabolic pathways, so don't worry too much about what that actually means. Just know that through metabolic pathways, we can release free energy by breaking molecules into smaller units, or build up molecules using smaller units by using energy. Okay, the difference between an autotroph and a heterotroph. Autotrophs, they use light, okay? If it's a photoautotroph, chemoautotroph use inorganic chemicals, and their carbon source is CO2, compared to heterotrophs, where we have photoheterotrophs use light. Chemoheterotrophs use organic compounds. Okay, their carbon source are also organic compounds. So what do you actually need to know? Autotrophs use light and inorganic chemicals as their energy source, and their carbon source is CO2, compared to heterotrophs, which use light and organic compounds, and their carbon source is organic compounds. Why do you need to know this? Well, this comes down to the structure of different microorganisms, okay? So photosynthetic prokaryotes are autotrophs, and then certain prokaryotes, such as Rhodobacter, are heterotrophs. So a typical question you might be asked is differentiate between autotrophs and heterotrophs, and you will need to say autotrophs can be photoautotrophs, which use light as an energy source, and chemoautotrophs, which use inorganic chemicals as an energy source. Both of them use CO2 as their main carbon source, compared to heterotrophs, which blah, blah, blah. Okay, gram-negative versus gram-positive bacteria. This is a different concept where we have two different types of bacteria. They're both bacteria, as the name kind of suggests. Gram-negative, however, have a double-layered cell wall, a very thin peptidoglycan layer, and lipopolysaccharide layer. Okay, a peptidoglycan layer is a polysaccharide. Okay, poly meaning many, saccharide meaning sugar. So many sugars bonded together. So peptidoglycan is just a sugar layer. And and that's for that one. A lipopolysaccharide is a molecule with both lipid, which is fat, and sugar. Okay, so fat and sugar together. Lipo means lipid, which means fat. Poly means many. Saccharide, again, means sugar. So fat with many sugars. Okay, again, that's why I like science, because you can just look at the prefixes and the suffixes and kind of work it out. Okay, so gram-negative have a double-layered cell wall, have a very thin peptidoglycan layer, and they have a lipopolysaccharide layer. It dyes pink or red when we dye it. Okay, when we put dye in it, it will dye pink or red. Gram-positive have no cell wall, or they'll only have a single cell wall. They have a very thick peptidoglycan layer and no polysaccharide layer. Okay, and they dye violet or purple. So these are two different categories of bacteria. So this is just a table that breaks it down. Cell wall in gram-positive, they might have a single cell wall, as I said, or they might not have any. Gram-negative are double. Gram-positive, they are more thick, and then gram-negative, it's thinner, because they have two of them, whereas because gram-positive only has one cell wall, it needs to be thicker. Peptidoglycan layer, they have a thick layer in gram-positive bacteria, whereas gram-negative have a thin layer. Okay, and you can kind of take a screenshot of the rest. This is what I was talking about. Okay, gram-negative, you can see that lipopolysaccharide layer that's lacking in the gram-positive. And you can see the peptidoglycan layer is thin, whereas in gram-positive it's nice and thick. And then yeah, you don't kind of need to know the other things apart from these cell walls. The fact that in gram-negative you can see there are two cell walls, where in gram-positive you can see there's only one. All right, let me just make sure there's no questions about that. No? Okay, cool. All right, because gram-negative bacteria have an outer layer, okay, so gram-negative have two outer layers, that protects them from their environment. Okay, so that external, again, Siri. Okay, sorry. Because they have two layers, it gives them more protection to their environment, as well as this lipopolysaccharide layer. So because of that, these bacteria can use this membrane to selectively keep antibiotic drugs from entering and killing them. Okay, so gram-negative bacteria are very protected against antibiotics, because remember, the way antibiotics work against bacteria is they come into the bacteria and effectively kill it. Okay, yeah, so it can't enter if it has this, or it's less likely to enter the antibiotic if it has this double-layered cell wall. This is a major issue for us because this means that more and more bacteria are becoming increasingly resistant to antibiotics. This means bacterial illnesses, once curable with antibiotics, will become less curable. Okay, that whole idea of antibiotic resistance. Hopefully we all remember that from high school. So, how do we combat that? Let's mock a tutorial for microbiology. I want to give you a taste of what a tutorial looks like. So let's imagine that was a small lecture, right? I was your lecturer, I told you all of the content. Now I'm your tutorial tutor. Imagine you've just entered your tutorial. The question is, could lactoferrin be the answer? This is lactoferrin here. What is lactoferrin? It's a protein found in breast milk, okay? It's iron chelating. What does iron chelating mean? It steals iron, it chelates it, it wants it for itself, okay? So if we put it next to bacteria, it will steal iron. Why do bacteria have iron? Because they need it for this. They need it for that membrane, okay? Bacteria need iron for their membrane. So if we introduce lactoferrin in an environment where bacteria is, gram-negative bacteria, we can deconstruct that cell wall. We can take the iron from the cell wall and then its cell wall will start diminishing. So antibiotics can then get in there and kill the bacteria. Okay, so that's why there's been plenty of studies into combining lactoferrin with antibiotics and getting the antibiotic in there more easily. This is a big area of research that is really probable for the combating antibiotic resistance. Okay, so Siri just keeps coming up. Lactoferin's a protein that could really help combat, and it's found in breast milk. Okay, so we've just mimicked what you do in uni. All right, we looked at the content, we went through it, we understood it, we did the lecture. Then in our little tutorial just then, we looked at it in the context of a real world problem, that problem being antibiotic resistance, and did some investigating on how we can combat this dangerous physiology of bacteria through the use of lactoferrin, okay? A side little note on lactoferrin and its use with antibiotics is we've actually seen in some studies bacteria becoming resistant to the lactoferrin. So it can still definitely be used and we see it be really, really successful. Unfortunately though, some bacteria are starting to defeat the lactoferrin and able to hold on to their iron really well. So the lactoferrin can't steal it from the bacteria. So yeah, there's still troubles with this, but it is still a really compelling piece of research. Okay, all right, so any questions on anything I just said or did or anything like that? Let me just check. No, okay, cool. All right, so let's go over my tips. First tip would be check Quizlet for your course or similar microbiology courses. If you type in your course name, so MICR 1001, for example, if that's the name of your microbiology unit, type it into Google and put Quizlet after, you will very likely find other people's Quizlets where they have flashcards on the course and the content. Also, another thing is a lot of your tests, exams, and quizzes have used questions that other unis have used and other years of your uni have also had. So you can likely find similar exam questions to what you will be asked online as well. Yeah, really, this is a tip for any course. It's not just exclusive to microbio only. Other tips: ask questions in tutorials and labs. Don't forget your tutors aren't just there to run the lab or tutorial. You can ask any questions you might have on the content, even if it has nothing to do with the tutorial lab to your tutor, okay? Make sure you ask questions about anything so it doesn't drag you down after when you do your exams. Yeah, this can really help ensure you have no confusions with the content. Look at your content outcomes. As I said, this is something really important. Often your content outcomes will be questions. As you go through them, as you go through the term, see if you can answer these questions confidently and correctly. Because there's no answer bank for those questions, it's easy to say, oh yeah, this is this, but make sure you actually check that that's the correct answer for the question. If they're not questions, simply phrase them into questions and do the above.